my YouTube channel. Today we will learn about conventional cloning. So first, let's understand what does gene cloning means. So gene cloning is the production of similar DNA in terms of characteristics and properties. So essential materials of cloning: target DNA, the specific gene or DNA fragment to be cloned. Then we need restriction enzymes to cut DNA and plasmid at specific sequence. Then we need vector like plasmid. And apart from plasmid, there are many kinds of vectors that are used in cloning like phagamid, yeast artificial chromosome, bacterial artificial chromosome, a carrier molecule, often a circular piece of DNA from a bacteria in case of plasmid, that can hold the target DNA and replicate within the host. Then we need ligase enzyme to join the target DNA with the vector. We need host organism, so in case of prokaryote like bacteria, E. coli, or in case of eukaryotes like yeast cells, mammalian cells, plant cells are used. Then we need growth medium. So why cloning vector? Cloning vector is a vehicle to artificially carry foreign genetic material into another cell where it can be replicated and expressed. It is used to amplify a single molecule of DNA into many copies. Cloning vectors are DNA molecules that are used to transport the clone sequence between biological hosts and test tube. So in the lab in vitro condition, we incorporate our gene of interest within a plasmid, and then this plasmid is incorporated within a specific bacterium using transformation. So without cloning vector, molecular gene cloning is totally impossible. So now let's discuss about one of the cloning vector, that is plasmid. Plasmid is an autonomously replicating circular double-stranded extrachromosomal DNA which is physically separated from the chromosomal DNA. Most general plasmids may be used to clone DNA insert up to 10 kb in size. Many plasmids, so plasmids are of two types. One is high copy number plasmid and another is low copy number plasmid. High copy number plasmids are used when you need to amplify your gene of interest or you need many copies of your gene of interest. On the other hand, low copy number plasmids are used when you need to express your gene of interest or you need proteins from your gene of interest. But high copy number plasmids should not be used for the protein expression because high copy number plasmid imposes a huge metabolic burden to the cell, and for that most of the protein will go to inclusion body, and it is very difficult to purify the protein or to solubilize the inclusion body in order to get our protein in solubilized form. So the example of plasmids like PBA322, PUC18, etc. So this diagram depicts the basic structure of plasmid. This is low copy number plasmid, that is PBA322, and this is high copy number plasmid, that is PUC18. So first let's understand about the naming of the plasmid. So this small b stands for the plasmid and BR stands for the Bolivar and Rodriguez, and 322 is its construction number because during the construction of the plasmid the scientists have constructed many different variants of the plasmid, and this is the construction number. And also for the PUC 18 or 19, this P stands for plasmid, UC stands for University of California, and 18 or 19 is the construction number. So both plasmid contains origin of replication from where the replication starts, then antibiotic resistance gene. For this PBA322, it contains ampicillin resistance gene as well as tetracycline, and for the PUC18, it contains ampicillin resistance gene, and also this plasmid contains MCS site or multiple cloning site. So in this site, many different types of restriction enzyme recognition sites are present, and this is the MCS site of this plasmid. So we used to ligate our gene of interest within this MCS site by cutting our gene of interest and also the vector with the same restriction enzymes. So why plasmids are good cloning vectors? Because of the small size, easy to manipulate and isolate, circular, so it's more stable, then replication independent of the host cell because it is extra chromosomal DNA, Then several copies may be present which facilitates in replication, frequently have antibiotic resistance gene that is used for the detection of the transformants. Disadvantages using plasmid, it cannot accept large fragment so the size limit is up to 10 kb. So now let's look into the basic steps of cloning. So this is our gene of interest and this is the plasmid, and in this case we have taken PUC19 plasmid. So we have cut our gene of interest with two different restriction enzymes, PSD1 and SAL1, and we have to cut this plasmid with the same restriction enzyme. So this is the PSD1 restriction enzyme recognition site and this is the SAL1 restriction enzyme recognition site. So after cutting our gene of interest as well as the vector with two different restriction enzymes, we need to ligate our gene of interest within the vector in order to produce recombinant DNA molecule. So there is a problem in ligating our gene of interest with the vector by cutting the gene of interest as well as the vector with only one restriction enzyme. So this is our vector and this is our gene of interest, and the MCS site or multiple cloning site is shown as black. So after cutting our gene of interest as well as the vector with only one restriction enzyme, sticky ends have been generated. So after ligation, this is our gene of interest and this is our vector. And this arrow shows that this is the promoter site. So RNA polymerase will start transcribing from this site and mRNA will be generated as CUG orientation. So in this case, this upper strand or red strand will act as a template, and from this this mRNA will be transcribed in CUG orientation, and from this mRNA further protein will be translated. But if our gene of interest is ligated with a vector in a reverse orientation, so in that case this below strand or this blue strand will act as a template. So in that case mRNA will be transcribed as GAC orientation or in a reverse orientation. So from this mRNA, altered version of a protein will be translated or no protein will be translated. And another problem of cutting our gene of interest and the vector with one restriction enzyme is that the vector can be self-ligated. So to avoid this problem, we need to cut our gene of interest as well as the vector with two different restriction enzymes, and this is called directional cloning. So once the recombinant DNA molecule is synthesized, then we need to transfer this into a host cell by transformation process. So once this recombinant DNA is transported within the host cell, multiplication of the recombinant DNA molecule will occur, and then division of the cell or division of the host cell will occur. Then we need to plate the cells over the agar plate and numerous cell divisions resulting in a colony. So how the selection will occur, or how can we select the cells that have the plasmid, or how can we select the transformants? So as you can see, this is our vector that contains ampicillin resistant gene. So after transformation, we will plate the cell over the agar medium that contains ampicillin. So only the transformed cell that contains the plasmid with ampicillin resistant gene will grow over the agar plate containing ampicillin, because the bacteria itself is ampicillin sensitive. So by this way, we can select the cells that contains the plasmid or the transformed cells. The non-transformed will not grow over the agar plate containing ampicillin. But this selection step doesn't ensure that the plasmid contains our gene of interest or the plasmid is recombinant plasmid. In order to select for the recombinant plasmid, we need to do blue-white screening, or we can do colony PCR from the colonies that will appear over the agar plate. So we can do colony PCR in two ways. One by gene specific primer, or another by vector specific primer. So for vector specific primer, the forward primer will bind here, this PUC forward, and the reverse primer will bind here, this PUC reverse primer. So if our gene of interest is not present, then after agarose gel electrophoresis, we will get a band to near to 136 base pair, the 720 minus 584, near to 36 base pair. On the other hand, if a gene of interest is present within this MCS site, then we will get a bigger size band. And the details of blue-white screening we will discuss in our upcoming video. Thank you.